Hello, this is Allison and Lauren, and we are here to take you step by step on our journey through discovering the real identity of our unknown bacteria D. We generated a dichotomous key based off of the chart that was provided to us, which included test results of all potential candidates for our unknown. We started off with the most basic classifications tests, which were gram staining and morphology of our unknown.
From there, we used our dichotomous key to determine which test would help us figure out what our unknown bacteria is. Using this method, we ended up performing the following metabolic tests: phenol red, TSI agar, catalase, oxidase, SIM, urease, and gelatin hydrolysis. Then, following all the metabolic tests, we performed different microbial growth tests to categorize the growth features of our unknown.
All of the tests we performed, we followed standard procedure so that we wouldn't contaminate our tests and get false results. For gram staining, we used a sterile inoculating loop to smear our unknown bacteria onto a slide. Then we heated the slide. After the bacteria was dried, we added crystal violet, iodine, ethanol, and safranin. After further examination under our microscope, we could determine that our unknown was gram-negative and bacillus-shaped. Due to our bacteria being gram-negative, then we can say that the crystal violet didn't soak into the thin peptidoglycan layer because it's too big, whereas the safranin is weak and water-soluble and therefore can soak into the thin peptidoglycan, making the bacteria appear reddish-pink. For the phenol red test, we obtained 3 different test tubes filled with phenol red glucose, sucrose, and lactose. Through a sterile procedure, we inoculated our unknown into each of the test tubes and incubated them at 30 degrees Celsius for 24 hours. The first test we performed was the gram staining. It showed a pink reddish color and rod-shaped bacteria, which allowed us to conclude that our unknown was gram-negative and bacillus-shaped. The second test we ran was the phenol red test with sucrose, lactose, and glucose. All 3 of these showed the same results, that there was positive color change and gas production. The color change indicated that there was acid production due to metabolism and fermentation of sucrose, lactose, and glucose. The gas production shown within the durum tubes means that there was gas produced during fermentation.
In the TSI agar test, we obtained a test tube filled with TSI agar, and then through a sterile technique, we inoculated a needle of bacteria into the agar. We then incubated the agar for 24 hours at 30 degrees Celsius. The third test we conducted was the TSI agar test. This test came back positive for glucose, lactose, gas production, and sulfide production. The slant and the butt of the test tube turned yellow, which indicated that glucose and lactose were fermented. There were bubbles present throughout the tube, which means that there was gas production during fermentation. Lastly, there were black growths throughout the tube, showing us that the unknown can reduce sulfur to produce iron sulfide. For the catalase test, we colonized our bacteria on an agar plate, and then using sterile technique, we picked a colony off of the plate and spread it on a glass slide and dropped hydrogen peroxide onto it. For the oxidase test, we used a sterile cotton swab to swipe bacteria off the agar plate. We then dropped oxidase reagent on the cotton swab and observed.
The fourth test we did was the catalase test, which came back positive because there were bubbles that appeared, meaning when we added hydrogen peroxide to our unknown, the hydrogen peroxide was broken down into H2O and O2. The O2 was released through the bubbles that were formed. The fifth test we ran was the oxidase test, which came back negative. When we placed the oxidase reagent on our unknown, it didn't turn purple, meaning our unknown bacteria didn't produce the enzyme cytochrome oxidase. For the SIM test, we obtained a SIM medium tube, and through sterile technique, we inoculated a needle with our bacteria straight down into the medium. Then we incubated the agar at 30 degrees Celsius for 24 to 48 hours. After being incubated, we dropped indole reagent on the top of the medium to see if there was a color change.
The sixth test we did was the SIM test, which was positive for sulfur reduction and motility, but was negative for indole. There was a black growth that appeared at the site of inoculation. Because it turned black, we can conclude that it was positive for sulfide reduction, which means that it produces its own energy by reducing sulfur to hydrogen sulfide. We know it is positive for motility because the black growth grows bigger towards the butt of the test tube. A positive motility test shows us that our unknown can move spontaneously, consuming energy in the process. We know that our unknown is negative for indole because when we placed the indole reagent on the top of the agar, it didn't create a pinkish film. This means that it can't hydrolyze tryptophan. For the urease test, we obtained a urea broth test tube, and using sterile technique, we inoculated a loop of our bacteria into the test tube, and then we incubated the test tube for 24 hours at 30 degrees Celsius. For the gelatin hydrolysis test, we acquired a test tube filled with nutrient gelatin. Then, using sterile technique, we inoculated the needle with our bacteria through the gelatin. We then incubated at 30 degrees Celsius for 48 hours. After incubation, it had to sit at room temperature for 30 minutes to see if the gelatin liquefied or solidified.
The seventh test we performed was the urease test, which came back negative. In other words, there was no color change, so we know that our unknown doesn't convert urea to ammonia or carbon dioxide. The final test we did was the gelatin hydrolysis, which also came back negative. When we tipped the tube upside down and side to side, it stayed a solid, whereas a positive test would turn to a liquid. This means that our unknown doesn't have gelatinase present. After conducting all of the metabolic tests, we determined that our unknown was Citrobacter frundi.
After doing all of our tests and receiving our results, this is a final run-through using our dichotomous key as to how we got to which unknown was ours. First, we started with the question, is the bacteria gram-positive? The answer was no. Is the morphology cocci? The answer was no. Is the O2 requirement obligate aerobe? The answer was no. Is phenol red test positive for sucrose? The answer was yes. Is the TSI agar test positive for hydrogen sulfide production? The answer is yes. Is phenol red test positive for lactose? The answer is yes. Is motility in SIMTest positive? The answer is yes. This way we were able to conclude that our unknown bacteria is Citrobacter frundi.
To test the growth of Citrobacter frundi in different pH levels, first we diluted a mixture of broth with our bacteria, which was a 1 to 50 dilution. Then we added 50 milliliters of our diluted bacteria into 6 test tubes of different HCl or NaOH concentrations, which was the acid in the base. The pHs ranged from 4 to 9. Then we incubated at 30 degrees Celsius overnight. We then tested each of the mixtures in a spectrophotometer to record how much bacterial growth of Citrobacter frundi there was at the different pH levels. To test the growth of Citrobacter frundi in different concentrations of NaCl, we took the same dilution of our bacteria, the 1 to 50 dilution, and added 50 milliliters to 6 different tubes of NaCl concentrations, ranging from 0 to 10. Then we incubated overnight at 30 degrees Celsius. Then we tested each mixture in a spectrophotometer to record how much bacterial growth of Citrobacter frundi there was at the different NaCl concentrations. To test the temperature tolerance of Citrobacter frundi, we used our 1 to 50 dilution of our bacteria that we used for the pH and the NaCl, and then added 50 milliliters to 6 different test tubes. Then we incubated these test tubes for 24 hours, each at a different temperature. The first one was put into 4 degrees Celsius into the fridge. The second was 22 degrees Celsius in room temperature. The third was 30 degrees Celsius in an incubator. The fourth was 37 degrees Celsius in an incubator also. The fifth test tube was put into 45 degrees Celsius into a water bath. The last one was at 60 degrees Celsius in a water bath also. Then we tested each test tube in the spectrophotometer to record how much bacterial growth of Citrobacter frundi there was at the different temperatures.
After we resolved what our unknown was, we performed various growth tests in different environmental conditions. We tested the growth of Citrobacter frundi in different pH levels, different NaCl concentrations, and different temperatures. We can interpret from our results that Citrobacter frundi is a neutrophile because it grows best between the pHs of 5.5 and 8.5. We can also determine that Citrobacter frundi is halotolerant because our results show that it grows best at no concentrations of salt, however still has moderate growth at low concentrations of salt. Lastly, from our results, we can see that Citrobacter frundi is a mesophile because it grows best at room temperature.
For the thioglycate test, we obtained a test tube filled with thioglycate, and without swirling or mixing the tube, we inoculated our bacteria straight to the bottom of it and incubated overnight at 30 degrees Celsius. Finally, we tested Citrobacter frundi to see if it grows best, or in some cases at all, in the absence or presence of oxygen. Looking at our results, we observed that Citrobacter frundi is a facultative anaerobe because there was an equal amount of growth that occurred throughout the whole test tube. Therefore, it can survive in an environment with or without oxygen.
We can conclude that Citrobacter frundi is a motile, gram-negative bacillus bacteria that is a facultative anaerobe, which tells us that it can perform aerobic respiration in the presence of oxygen, but it also has the ability to switch over to fermentation and anaerobic respiration in the absence of oxygen. We can back this up through the metabolic tests that were positive for sulfur reduction and sucrose, lactose, and glucose fermentation. The catalase test shows that the enzyme catalase can protect the bacteria from toxic byproducts of oxygen metabolism when the bacteria does perform aerobic respiration.